I'm going to talk about potential for diagnostics based on the microbiota, potential for therapeutics, harnessing new members of the microbiota, and I'll talk about some small molecules which can be harvested from the microbiota and used to potentially cure diseases. And the paradigm which I'm putting forward to you is that in the future, when a patient goes for consultation, as well as having their blood pressure checked and the normal anthropometrics, they will undergo microbiome profiling. And if their microbiome suggests there are risk factors there, they may be administered live biotherapeutics or small molecules derived from the microbiota. I predict that your microbiome profile will become a part of your health records, and it will be checked annually, and it will be used to check risks for various diseases. Those diseases could be, as we heard this morning, risk for childhood obesity. It could be risk for frailty in old age. It could be drop in microbiota diversity and susceptibility to Clostridium difficile. A graphic example of how this works is an example from the work of Duško Ehrlich and colleagues in France in the so-called MetaHit project. And this graph is an illustration that people who are obese, have a BMI above a threshold, have a lower microbial gene count in their gut than either the entire cohort or healthy people. So at face value, the observation is if you are obese, you have fewer microbial genes in your microbiota. That's an interesting phenomenon. It transpires that you can detect this low gene count phenotype. You don't have to sequence the whole microbiota. You can look at the prevalence levels of 19 species. And the important thing is that if you try and make these obese people lean, the lower the gene count, the less responsive they become. So you can stratify obese people into low gene count and really, really low gene count and predict if they're going to be able to respond to therapy, which could be caloric restriction or a combination of diet and exercise. Another example I'm going to quote from my own work is irritable bowel syndrome. And one of the most difficult aspects of IBS is that the combined therapy response rate is about 40%, but the combined placebo response rate is about 30%. So treatment works 10% better than placebo, which is really a terrible statistic. We started to study the microbiota in IBS. Our current working hypothesis on IBS is that some of the patients have an altered microbiota and they have a true functional gastrointestinal disorder based on that microbiota. They have a lower luminal pH. They've got an overabundance of phylum Firmicutes. And these are probably the patients who would respond to a FODMAPs diet, which is removing fermentable carbohydrates from their diet. The interesting clinical feature of the IBS patients with a normal microbiota is that they have much higher scores for anxiety and depression. And psychiatric comorbidities are a typical feature of some but not all IBS patients. So our hypothesis for those subjects is that they have a functional gastrointestinal disorder because the brain-gut axis is working in a downward direction and there are disturbances from the brain working down. Another condition that we're interested in is cancer. It began to be reported in the literature that the microbiota on colon cancer tumors was different to the normal microbiota. We started to look at the microbiota in colorectal cancer in subjects in Ireland. What we discovered is that the microbiota on the tumor at surgery is different from the typical colonic microbiota of healthy people who presented for colonoscopies. Interestingly, we also looked at a bunch of polyps, which are small, benign growths which may grow to form full tumors. And they had a microbiota which was intermediate between the controls and the colorectal cancer microbiota, which fits with the notion that there's a transition in microbiota from that in healthy people to that in colorectal cancer. We were able to subdivide the colorectal cancer microbiota into 6 types. 2 of those types had a relatively low risk, but 4 of them had a much higher risk for colorectal cancer. And in this gut paper, which you can read if you're interested, we were also able to correlate the microbiota subtype with the host response based on a transcriptome analysis of the tumor biopsy. So what that means in practice is that different colorectal cancer patients have different immune responses. They're crudely grouped into Th1 or Th17, and they've got much different clinical prognoses. And we were able to predict what the host response would be based on the adherent microbiota. What about therapeutics? One of the notions which has come out of this research is that there must be other beneficial bacteria in the gut, and by studying the microbiome, we can identify them and use them as therapeutics. Fecal microbiota transplantation, as we just heard, is wonderfully effective for C. diff. It actually works very poorly for inflammatory bowel disease, for IBS. The idea is let's use FMT as a paradigm and drill down into the microbiota and try and identify new organisms. And Miguel also referred to this company on the east coast of the US, Series Therapeutics. They've received incredibly large amounts of money in IPO investments. Their lead product, Series 109, is for recurrent C. diff, but they have a pipeline of products which are hopefully going to be more defined than just spore cocktails for conditions like primary C. diff and for a variety of other diseases. Another example which I really like is based on work by Willem de Vos, who is in Wageningen in Netherlands and Helsinki in Finland. And Willem was also working on FMT, but he was doing FMT for metabolic syndrome. And what they noticed that in the patients who responded well to FMT and metabolic syndrome, one of the organisms that transplanted the best was Eubacterium hallei. At the same time, they had an animal model where they treated rats with vancomycin and essentially caused them to have metabolic syndrome. And one of the organisms that was killed the most was Eubacterium hallei. And indeed, there's an explosion of startup companies in this area. The top half of the slide is an example based on work by David Mills, Lebria and colleagues in UC Davis in California. And they are world experts in understanding why breast milk is so good at promoting certain classes of bifidobacteria. It's only some subspecies and some strains of bacteria which respond to the human milk oligosaccharides in breast milk. And they are trying to use this information to develop better prebiotics for human infants to allow them to develop a microbiota which is more like what the gold standard would be if we were all receiving, as infants, human breast milk. A similar example using a different approach is a company called Commence. And based on the observation, and I showed you some data earlier on, that C-section infants have a very different microbiota. And based on the hypothesis or the fear that they will have diseases later in life because of that, the idea here is to inoculate the infants with normal vaginal microbiota. It's an interesting concept. It's going to be very challenging because young babies are obviously extremely at risk. And if you think about what you're doing, you're changing the course of their microbiota for the rest of their lives. I predict that microbiota profiling will become part of your diagnostics and your medical records. So if that's true, it means that the primary healthcare providers will first of all have to have mechanisms for collecting samples. They'll have to have good liaisons with sequencing labs. And the primary physicians or the nurses will have to be able to communicate microbiota data. So we have to be able to translate findings into actionable recommendations for obesity in children or in older people or type 2 diabetes or whatever. It has also been shown that it should be possible to identify individuals based on their microbiota fingerprint. So any primary healthcare facility with microbiota data will have to have the same standards for data security and confidentiality as they would for human genome sequence data or any other sensitive clinical data. Therapeutics, the director of my research institute distinguishes between the broken biome and the broken host. A good example of this is IBD. Inflammatory bowel disease could be simplified as an inappropriate immune response to an appropriate microbiota component. And it has genetic parts and environmental parts. And there are a large and growing number of mutations or SNPs associated with IBD. I would characterize IBD as a broken host more than a broken biome. And if the host has strong genetic components in the disease, you have to be realistic about the potential for giving clinical amelioration or cure in that case. However, biomarkers may still be useful. So even in the example of IBD, it could be that the microbiota changes can be used to detect a flare-up, and management of the patient can be handled accordingly. So microbiota management and restoration seems to be a useful area in, for example, frailty. One of the interesting applications of FMT is that there seems to be prospects for controlling carriage of multi-drug resistant organisms. Already people have developed and patented genetically modified organisms for human therapeutics. A good example is the work by Lothar Steitler, who previously worked in a company called Actogenics. And what he developed was a Lactococcus lactis expressing human IL-10. And this was shown to be therapeutically helpful in patients with IBD. And his company has been bought out and has massively expanded its range of products. But the common technical platform is the same. These are human commensal organisms, or lactic acid bacteria, and they're GMOs. They're expressing human proteins which are typically anti-inflammatory. This is never going to be an over-the-counter solution. These are going to be prescription medications for people who are experiencing severe disease, but it's still an exciting example of a microbiota-based therapeutic. We have to be cognizant of issues of ownership. If I recruit a patient, and if I isolate a strain from that patient, which is clinically and commercially very useful, who owns it? It depends on the consent form which the patient signs. But these are issues for commercialization. You also have the issue of the Nagoya Protocol, which is a global agreement whereby indigenous people will not be studied, sampled, and microbes taken from them. So all strains used for commercialization in signatory countries to the Nagoya Protocol must be compliant with that in terms of informed consent from the donors of the microbes. And we're probably going to need a new kind of regulation for live biotherapeutic products. The last slide is just a reminder that we all have slightly different genomes, and we all have different microbiomes. And evidence is emerging from initially animal studies and now human studies that there is a genetic component to the microbiota that you carry. Some of the polymorphisms are to do with innate immunity genes, which regulate how you control your microbiota. So it may transpire in 10 years' time that it's not possible to introduce very different organisms into particular groups. That's why I think it would be fascinating to know what is the typical Mexican gut microbiota. I don't think that question can be answered today. We have got great data for North Americans and Europeans, but I think once we finally have large microbiome data sets from lots of different countries with well-known human genomes, we'll begin to understand these subtle human genome, human microbiome interactions. Thank you very much.